Hello and welcome to the wonderful world of science. I'm Darion Donnelly. Early this morning, scientist and researcher Doubles McGee successfully, or so we thought, cloned what started out as a puppy dog. For those of you unfamiliar with cloning, there are three different types currently in existence. They are reproductive cloning, therapeutic cloning, and DNA cloning. Here's Doubles McGee with more.
Some of you may be familiar with Dolly, the famous sheep who was successfully cloned. But what does it actually mean to be cloned? Cloning is an intricate process stepping over some moral boundaries. It involves duplicating an individual. Basically, the clone will have the same genetic makeup or DNA as the original. However, to describe cloning, it is important to note that there are several different types. These include reproductive cloning, therapeutic cloning, and DNA cloning. Within reproductive cloning, scientists use somatic cell nuclear transfer, or SCNT. The genetic material is taken from an adult nucleus cell and transferred to an egg without a nucleus or genetic material. Cell division is then stimulated, and at the right stage in development, the cell is transferred back to the host in the uterus for birth. Therapeutic cloning may be more recognized as embryo cloning. Basically, embryos may be cloned for research. The benefits of this type of cloning may be finding medications, cures for diseases, and studying the development of human beings. With stem cells, scientists can create any cells within the body to experiment with. The stage in which the embryo is disturbed is called the blastocyst, development stage of the egg. Gene or DNA cloning consists of DNA fragment transfer from an organism to another element, a bacterial plasmid for example. The DNA will be put into a host cell. This process is extremely prevalent in molecular biology in many labs since the 1970s. In this case, I used reproductive cloning. The clone, who I have named Napoleon the Great, has remained in his cage since birth. Here he is now. Wait, what's that? The cage is unlocked?
Well, it looks like her experiment didn't go as planned. More on that later. But thank you for tuning in to the currently not so wonderful world of science, and remember, don't try cloning at home.
I would like to thank my sources, the U.S. Department of Energy Genome Program, as well as Dr. Gruse's genetics class that I took last year. Also, I would like to thank my boyfriend, Darion Donnelly, for performing so well, and my dog Maya, who is not a crazy killer.